Dear students, today we are going to study one of the most important organelle of cell, that is nucleus. There are certain organisms in which nuclear material is not bounded by nuclear membrane. They are placed under the category of prokaryotes. Their nuclear material is referred to as nucleoid. Hence, the nuclear membrane is absent in prokaryotes, but those organisms in which the nuclear material is bounded by the nuclear membrane, or we can also say that those organisms which have nucleus, they are placed under eukaryotes. This nucleus was first of all discovered in 1831 by Robert Brown. As far as its shape is concerned, it can be spherical, rounded, ovoidal, prismatic, elliptical, pyriform, or it can also be in lanceolate form. Come to the structure of the nucleus. There are two parts present in the nucleus. One is nuclear envelope, and the part which is contained inside the nuclear envelope, that means the nucleoplasm. The nuclear envelope is a double membrane structure separated by perinuclear space. Frequently it is perforated by pores. Each pore has a ring or cylinder called as annulus. The nuclear membrane has two trilaminar unit membranes of 7 to 8 nanometer thickness, and this outer membrane communicates with the endoplasmic reticulum many a times. It has also ribosomes attached to it on the outer surface, and it displays the phenomenon of blabbing, that is formation of vesicles. After the outermost nuclear membrane, the outermost nuclear membrane is followed by perinuclear space. And what I wanted to say is there are two trilaminar unit membranes, and between them is a place called as perinuclear space. This perinuclear space is around 20 nanometer in size, and it is filled with fluid. The nuclear membrane is frequently perforated by nuclear pores, and which are circular or octagonal in shape, occurs in clusters, may be arranged in rows, or they are randomly scattered. The nuclear pore, they contain annulus, which is electron dense cylinder within the pores. It has 8 subunits. If we cut the section through the nuclear pore, it shows presence of an electron dense cylinder within pore. The actual opening of the nuclear pore is thus confined to the cavity of annulus. This annulus extends on both sides of the nuclear membrane, that is towards cytoplasm and nucleoplasm as well. Annulus consists of 8 subunits, as I have told you, arranged radially around the periphery of the pores. You can view the structure of annulus. Annulus is fitted as a cylinder in the nuclear pore. This structure is given by Wirsner, and you can see that 200 angstrom sized what we call it subunit is present. There are 8 such subunits, as I have told you in the beginning of the lecture, and these are arranged around a 500 angstrom space, which is the actual passage towards the nucleoplasm or from towards the cytoplasm. This annular material extends beyond the pore margins, and these pores are octagonal in shape. The annulus of pore is composed of amorphous matrix in which have granules and thin filaments, which have dimension of 40 to 70 angstrom, and the filaments are of the dimension of around 30 angstrom. You can also view another diagram of this pore complex. You can see the peripheral granules present, the outer nuclear membrane followed by perinuclear space, then the inner nuclear membrane which remains in connection with the nucleoplasm. The outer nuclear membrane remains in connection with the cytoplasm, and as you can view that there is a central granule, and central granules have filaments also, as I've told you. And these central granules, they are ribonucleoprotein particles because they are dissociated by RNAs. The central granule is around 10 to 15 nanometer. It's not a constant feature and is sensitive to RNAs, and thus we can deduce that it is made up of ribonucleoproteins. Following the innermost innermost membrane is fibrous lamina present on the inner surface of the nuclear envelope. Its thickness is around 280 nanometers. So this is all in the structure of the nucleus. And now let us come to the functions of the nuclear membrane. The prime function of nuclear material, this nucleus is it separates protein synthetic machinery from the genetic component of the cell. It protects the DNA against the mutagenic effect of cytoplasmic enzymes. It saves, so therefore we can assume that the genetic material of eukaryotes is stabler than that of prokaryotes, because a jacket of protection is afforded by the nuclear membrane, and the nuclear membrane prevents the DNA from the mutagenic effects of the cytoplasmic enzymes. Such a protection is absent in prokaryotes, therefore mutations are more frequent in prokaryotes. The greater stability of eukaryotic DNA is partially due to this, can be partially attributed to this nuclear envelope. Therefore, nuclear envelope can be called as a dynamic barrier. Then, nucleocytoplasmic exchange. The RNA which is formed by the DNA of the nuclear material is transported to the cytoplasm along with macromolecules, small organic molecules, waters and ions through nuclear envelope. Pores are the major site of transfer of these materials. The transport is effected by active transport also and passively also. Blabbing also occurs, which is the outpocketing of the nuclear membrane, and the material which is packed in the blabs is transported to the cytoplasm. Some material is also exchanged through endoplasmic reticulum. As I have told you that some endoplasmic reticulum remain attached to the nuclear membrane, outer nuclear membrane, and found in continuity with it. The nuclear membrane also provides the attachment sites to myofilaments, microtubules, and microfilaments. The nuclear component is attachment site to the centromeres, telomeres, to the nuclear envelope. Bar body synaptonymal complex, they remain attached to the nuclear membrane. Then the nuclear envelope or the nuclear membrane also contributes to other cell membranes by the phenomena of blabbing. As I've told you, the outer nuclear membrane gives off vesicle and contributes to the endoplasmic reticulum, which thereafter contributes to the Golgi membrane of Golgi bodies, and so on, and ultimately to the plasma membrane. Some electron transport activity is also demonstrated by the nuclear membrane, similar to that which is shown by endoplasmic reticulum. By presence, it shows by presence of NADH cytochrome C reductase, NADPH cytochrome C reductase, and flavoprotein cytochrome P450, which are similar in nuclear envelope also. The ribosomes are also remain attached to the nuclear membrane. That's why it also shows the activity seat of protein synthesis. So students, this is all in this lecture. We'll come up with nucleolus in the upcoming lectures. Thank you so much.